the International Atomic Energy Agency, IAEA. How is the COVID-19 virus detected using the real-time RT-PCR? March 27, 2020.
Good evening.
IAEA Office of Public Information. Nuclear explained. As the coronavirus that causes COVID-19 disease spread across the world, the IAEA, in partner with the Food and Agriculture Organization of the United Nations, FAO, offering its support and expertise to help countries use the real-time reverse transcriptions, polymerase chain reactions, real-time RT-PCR, one of the most accurate laboratory methods for detecting, tracking, studying the COVID-19 coronavirus. But what is real-time RT-PCR? How does it work? How is it different from the PCR? And what does it have to do with the nuclear technologies? Here is a handy overview of technique, how it works. A few the refreshes detail of virus and genetic. What is real-time RT-PCR? Real-time RT-PCR is a nuclear-derived method to detecting the presence of the specific genetic material in any pathogens, including the virus. Originally, method to use the radioactive isotope and measures, detect target genetic material. So, subsequent refining that has led to the replacement of isotopics labeling with the special markers, most frequently fluorescence D. This technique allows the scientists to see the result of almost immediately while the process is still ongoing, whereas conventional RT-PCR only provide the result at the end of the process. The real-time RT-PCR is one of the most widely used laboratory methods to detecting the COVID-19 virus. While many countries have used the real-time RT-PCR for diagnosing other diseases, such as Ebola virus, Zika virus, many need to support in adapting this method for COVID-19 virus, as well as increasing their national testing capability.
Thank you. Appreciate it.
What is virus? What is genetic material? A virus is microscopic package of the genetic material surrounded by a molecular envelope. This genetic material can be either deoxyribone nucleic, deoxyribone nucleic DNA, and the ribonucleic acid RNA, DNA and RNA. DNA is two-strand molecule that is found in the organism such as animal, plants, virus, which is hold the genetic code blueprint. All these organisms are develop, and RNA is generally one-strand molecule copies, transcript, and transmit part of a genetic code protein so that they are synthesized, carry out the function that keep the organisms alive, developing the different variations of RNA. So RNA are responsible for copy, transcribing, and transmitting. So some viruses, such as coronavirus, SARS-CoV-2, which is called COVID-19, only contain RNA, which is mean rely on infiltrating healthy cells to multiply and survive. Once inside the cell, the virus use all genetic code. The RNA is case of COVID-19 virus. They take control of reprogram the cells, turning them into the virus-making factories. In order for virus like COVID-19 virus to be detected early, the body using the real-time RT-PCR, scientists need to convert RNA. The scientists need to convert RNA to the DNA. This is process called reverse transcriptions. They do this because only DNA can be copied and amplified, which is a key part of the real-time RT-PCR process detecting the virus. Scientists amplified specific part of the transcribed by the DNA hundreds of thousands of times. Amplifications is important so that instead trying to spot minuscule amount of the virus among the millions of strange genetic information, the scientists have large enough quantities of target sanctions virus DNA to accurately confirm that the virus is present. So how does real-time RT-PCR work with the COVID-19 virus? A sample is collected from the part of body where the COVID-19 virus gathers, such as person nose, or throat. The sample is treated with the several chemical solutions that remove the substance such as protein and fats. Extract only RNA present in the sample. So the extracted RNA is a mix of the person's own genetic material and if present the virus RNA. So the RNA is reverse transcribed to DNA using the specific specific enzyme. Scientists learn additional short fragments of the DNA. They are complementary, specific part of transcribed viral DNA. If the virus is present in the sample, these fragments attach themselves to target section of the viral DNA. So some of the added genetic fragments are used for the building the DNA strands during the amplifications, while the others used to the building the DNA, adding the markers labeled to the strands, and which are used to detect the virus. So the mixture then placed in RT-PCR machine. The machine cycle through the temperature, the heat, and the cool. Mixture of the triggers specific chemical reaction not create new identical copy of the target sections of the viral DNA, and the cycle repeat over and over to continue copying the target sections viral DNA. Each cycle double previous number. 2 copy become 4, 4 copy become 8, so on. So the standard real-time RT-PCR setup usually go through the 35 cycle, which is mean by the end process, around the 35 billion new copy of the sections of the viral DNA created from each strand of the virus present in the sample. So a new copy of viral DNA sections are built, and the marker label attached to the DNA strands. So then release a fluorescence D, which is measured by the machine computer and present the real time of the screen. So the computer checked amount of fluorescence in the sample after each after each cycle. In order for the virus like COVID-19 virus to be detected early in the body, using the real-time RT-PCR, scientists need to convert the RNA to the DNA. This is a process called the reverse transcriptions. They do because the only DNA can be copied, amplified, which is key part of real-time RT-PCR process for detecting virus. The scientists amplified part of the transcript viral DNA hundreds of thousands of times. Amplifications is important instead of trying to spot minuscule amount of virus among the million of strands genetic information. The scientists have large enough quantity of target sections of viral DNA accurately confirm that the virus is present. So how does real-time RT-PCR work with the COVID-19 virus? A sample is collected from the part of the body where the COVID-19 virus gathered. So such a person knows throws a sample of treat with several chemical solution. Remove the substrate such as protein, fats, extra only RNA, present the sample. This extra RNA is a mix of person's own genetic material, the virus RNA. Why use the real-time RT-PCR? The real-time RT-PCR technique what is PCR and different from the let me speak. The International Atomic Energy Agency, IAEA. How is the COVID-19 virus detected using the real-time RT-PCR? March 27, 2020. IAEA Office of Public Information. Nuclear explain. As the coronavirus that causes COVID-19 disease spread across the world, the IAEA, in partner with the Food and Agriculture Organization of the United Nations, FAO, offering its support and expertise to help countries use the real-time reverse transcriptions, polymerase chain reactions, real-time